Hi, I'm Sandra Porter. I'm going to talk today about using the NCBI databases. So I've already gone to NCBI. The URL is ncbi.nlm.nih.gov, stands for the National Center for Biotechnology Information at the National Library of Medicine at the National Institutes of Health. To search the databases, I put whatever term I want to search for into the search box here. And I can just click return. And it's going to search all the databases at the NCBI at once. So I've searched for them all for ACE2. I can see kind of a summary for the gene information at the top. I can see some information about the RNA molecules that are made from this gene. And I see that there are all kinds of records in many of the NCBI databases. The numbers that appear here tell me how many records there are. So for example, in the PubMed Central database, this contains open access medical biomedical literature, 36,633 papers related to ACE2. And if I click this link, I can read some of them and read the abstracts and see who wrote them and what they wrote about. Down here in genomes, I could see there are 3 taxonomy references, 183 records in the genome database, over 3,000 records in the nucleotide database, 167 clinical trials. I can see information about genetic conditions related to ACE2, information about pathways, information about chemicals, information about protein structures, all kinds of things about the ACE2 protein. What we're going to look at right now, though, is we are going to look at the gene database, which I can see has 1,247 records. All right, so I click that to go to the gene database. The record at the top is human, so that's the one I want to look at. I'm in luck. I can get there right away. But I might also want to search, and it's not always, I'm not going to want to read 1,247 records to find the information I want. I can search the information on this particular page just by using the same command that I use to search in Word or other web pages or other things. I do Command F. It opens up a little tiny box down in the lower left-hand corner. And let's say I wanted to see about an ACE2 gene in a pig. I could type pig, and you see it went to it right here, and it highlighted the word pig. So that's pretty handy, a quick way to search, a quick way to find things. But right now, actually, I am going to look at human, so I'm going to type human in here. And I see the first record is human, and then I can also see it's human right here too. This gives me a little bit of additional information. I can see the whole name of the enzyme that this gene encodes. I can see that it's on the X chromosome. I can see an accession number for the sequence, for the chromosomal sequence. And now I'm going to click the record, or click the link and go to the record and learn about the gene record. Okay, so I can see that this gene codes for a protein. I can see the official name of the enzyme. I can see the lineage that made by eukaryotes, metazoa, chordata, and the rest. I can see a summary about the gene function, what's known about the gene function. I also have a table of contents over here that gives me additional information, which can be helpful for quickly navigating through the page. And I can scroll down and see other information as well. So right now, I'm going to go to the genomic regions, transcripts, and products. This menu right here tells me I am looking at chromosome X, and I'm looking at the reference sequence, and this is GRCH38.P13. that's the build number. So it tells me which version of the sequence I'm looking at. There are other kinds of sequences I could look at here as well. There is one of the ref-seq genes is here. There's also an older version of the human sequence, but we're going to stick with the default version 38. Okay, as I look down, we're going to look at the NCBI Homo sapiens version here. I can see there are, as you see, there's 6 green horizontal lines. 1, 2, 3, 4, 5, 6. And in each line, you see there are vertical lines, lots of them in some cases. Those are used to represent the exons. I'm going to zoom in here. I just press the little plus key. Okay, so those vertical lines, those are used to represent exons. Exons are the pieces of messenger RNA that are spliced together, or pieces of RNA, pre-messenger RNA, that are spliced together to create the messenger RNA. You can see, it's not so easy to see, zoom in a little bit more, that sometimes these exon boxes, or these exons, have different shading. So here you can see there's kind of, I guess we'll just look at this one. There's one that has kind of a lighter shade, and then a bit lighter shade, and then a darker green shade. Those parts of the boxes that have the light green shade, those are going to be parts of the messenger RNA that are parts of the messenger RNA, but they're not actually coding for protein. It is the dark green parts that actually code for protein. Okay. So we've got lots of different exons here, or lots of different transcripts, pardon me. Each RNA is referred to as a transcript because it's made through transcription. An RNA copy. Transcription is the process of making an RNA copy of DNA. And if I want to know more about any one of these, I can click it and learn more. So each of these has a number at the end, at the left end. That is called an accession number. It starts, these here all start with NM, and that number signifies that this is a reference sequence and that it is RNA. Okay, now I'm going to click this. And when I do that, it kind of opens up the information that belongs to that gene. And it shows me here is the messenger RNA below in purple. I'm going to zoom out a little so I can see this all at once. And below in red, I have the protein sequence that goes along with that messenger RNA. So you can see here the RNA begins here relative to the DNA, and the protein coding region doesn't start till over here. So it also shows you that there is a region of that messenger RNA that is a region of the RNA that is used in the messenger RNA that is not used in the protein. I can get other information as well if I'm very careful with holding my cursor. So I'm going to hold it carefully over the accession number, and I can see that my RNA sequence, right, where I can see that the whole DNA sequence, might as well start there, is 46,179 nucleotides. The RNA sequence is 3,135 nucleotides for this particular RNA. And again, this is the accession number, the number that refers to this RNA. If I want to know something about the protein, I can carefully hold my cursor over the protein sequence, the beginning of the protein, and I see that my protein, the coding sequence that codes for the protein, is 2,085 nucleotides, so not as long as the messenger RNA. It codes for 694 amino acids. And see, is there anything else I want to say? Oh yeah, and that this is the information, this is the accession number for that protein. Okay. Other information we have about this, we talk in the assignment about looking at expression. Expression is the process of making those RNA molecules from the DNA. And now, these days, we're pretty good at sequencing RNA. And so one thing people do is they sequence RNA from different tissues to understand more about where different proteins are produced, when different proteins are produced, and sometimes what versions, which transcripts are produced from different proteins. So we're going to look at the expression graph for ACE2. We've actually got 4 different graphs here that we can vary between. Here we have the graph of the RNA-seq data from normal tissues from a HPA, the Human Proteome Project, I think. We can see that this came from samples from 95 people, and they represent 27 different tissues. We could see when this was published, and we could actually go and look at the paper if we wanted to. Here, RPKM is a value that is used to compare the amounts of RNA between experiments, and that's defined in the assignment, so you can read a little bit more about it there. And we can see here the number of RNA molecules that are produced in all different kinds of tissue. So here in the colon, if I hold my mouse over this, it shows me that an average or a mean of 4.6 RNA molecules were found in the colon. Here, I can see that an average of 67.6 RNA molecules were found in the duodenum. Here I've got 2.3 molecules in fat tissue and 32 RNA molecules reads per RPKM per gallbladder, and so on. And if I want to look at different tissues, I select the different database or the different data set from the menu here. I can see the values again, and I could use these RPKM numbers to kind of help me compare between the different data sets. And that's enough for now, but there's a lot of information in a gene record. You can see why there's a table of contents above to help you get through it and help you find all these things.